Hello students, welcome to Read Med Prep Academy channel. Today in Molecular Genetics Part 6, we are going to discuss about genetic code, mutation and the genetic code and Wobble Hypothesis. What is the genetic material? DNA is the genetic material. It carries the genetic information in the cell from generation to generation. We are going to make an attempt to determine in what manner the genetic information exists in the DNA molecule and how the genetic information is coded in the DNA molecule as a coding language. If they occur in the language of codes, what is the nature of the genetic codes? Here you can see that the DNA undergoes replication on its own. When the DNA undergoes transcription, it forms the messenger RNA, and the messenger RNA causes translation of proteins. In some viruses, the RNA is the genetic material and the RNA forms the DNA. This is called reverse transcription. What is triplet rule? The translation of proteins follows the triplet rule. A sequence of 3 messenger RNA base, that is called a codon, designates one of the 20 different kinds of amino acids used in protein synthesis. Genetic code is the sequence relationship between the nucleotide in the genes or the messenger RNA and the amino acids in the proteins they encode. So what is the definition of a codon? It is a specific sequence of 3 consecutive nucleotides that is part of the genetic code and that specifies a particular amino acid in a protein or starts or stops the protein synthesis. This is also called a triplet. There are 64 possible triplets and 61 of them are used to represent amino acids. The remaining 3 triplet codons are termination signals for polypeptide chains. Since there are only 20 amino acids involved in protein synthesis, most of them are encoded by more than one triplet. Here you can see that a codon is made up of 3 messenger RNA nucleotides. There are 64 codons totally. AYUG is a codon that encodes the methionine and starts translation of all the proteins. 61 codons encode 20 amino acids. The redundant codon is GCA. The 61 codons are the codons that code for the proteins and 3 codons stop the protein translation. They are UAA, UAG and UGA. 2 things make this multiple degenerate coding possible. First, there is more than one transfer RNA for most amino acids. Each transfer RNA has a different anticodon. Here you can see the messenger RNA, it has a codon and the codon has an anticodon in the transfer RNA and the anticodon helps in bringing the amino acid to the messenger RNA. Second, this pairing is highly specific for the first 2 portions of the codon, permitting Watson and Crick base pairs, A to U and G to C to be formed. But at the 3rd position, there is a great deal of flexibility as to which base pairs are acceptable. Most part of the genetic code is universal, being the same in prokaryotes and eukaryotes. The order of base pairs along the DNA molecule controls the kind and order of amino acids found in the proteins of the organism. This specific order of base pairs is called the genetic code. The blueprint establishing the kinds of proteins to be synthesized which makes an organism unique. Marshall Nirenberg, Severo Ochoa, Hemoglobin, Corona and Francis Crick and many others have contributed significantly to decipher the genetic code. The enzyme polynucleotide phosphorylase called Ochoa's enzyme was named after Severo Ochoa, which helps in genetic coding. Now the order in which the bases are arranged in the messenger RNA decides the order in which the amino acids are arranged in the proteins. Finally, there is a checkerboard for the genetic code and this was prepared. Let us go to the checkerboard. You can see on the left side is the first letter. The table is UCAG and the second letter on the top which is UCAG and the 3rd letter on the right side. Now let us go to the checkerboard. You can see that the codon UUU, UUC codes for phenylalanine. UUA, UUG codes for leucine. UCA, UCC, UCA, UCG codes for serine. UAU, UAC codes for tyrosine. UAA, UAG are stop codons. UGU, UGC are coding for cysteine. UGA is a stop codon. UGG codes for tryptophan. Now coming to the second row, CUU, CUC, CUA, CUG codes for leucine. CCU, CCC, CCA, CCG codes for proline. CAU, CAC codes for histidine. CAA, CAG codes for glutamine. CGU, CGC, CGA, CGG codes for arginine. Coming to the third row, AUU, AUC, AUA codes for isoleucine. AUG codes for methionine. ACU, ACC, ACA, ACG codes for threonine. AAU, AAC codes for asparagine. AAA, AAG codes for lysine. AGU, AGC codes for serine. AGA, AGG codes for arginine. Coming to the fourth row, GUU, GUC, GUA, GUG codes for valine. GCU, GCC, GCA, GCG codes for alanine. GAU, GAC codes for aspartic acid. GAA, GAG codes for glutamine. GGU, GGC, GGA, GGG codes for glycine. So we must learn to use the checkerboard, and we must know how to formulate the checkerboard. It's not very difficult if you follow the first letter, second letter and third letter pattern. Now we must know about amino acids code. The amino acids are in full name and represented by 3 letters and are represented by a single capital letter. Now let us go one by one. Non-polar and uncharged amino acids. Alanine represented by ALA, capital letter A. Phenylalanine PHE, capital letter F. Glycine GLY, G. Isoleucine ILE, I. Leucine LEU, L. Methionine MET, M. Proline PRO, P. Valine VAL, V. Tryptophan TRP, W. Polar and uncharged amino acids. Cysteine CYS, represented by capital letter C. Asparagine ASN, N. Glutamine GLN, Q. Serine SCR, S. Threonine THR, capital letter T. Tyrosine TYR, Y. Now positively charged amino acids, otherwise called basic amino acids. Histidine HIS, capital letter H. Lysine LYS, K. Arginine ARG, R. Negatively charged amino acids which are acidic. Aspartic acid ASP, capital letter D. Glutamic acid GLU, capital letter E. Some ambiguous codes are asparagine or aspartic acid is represented as ASX, capital letter B. Glutamine or glutamic acid GLX, capital letter Z. Now what are the saline features of the genetic code? The genetic codon is a triplet code, and 61 codons code for the amino acids, and 3 codons do not code for any amino acid, and the function as stop codon, otherwise the termination codon. Here you can see the codons are sets of 3 RNA nucleotides that hold the code for an amino acid. The GCA codon on the left side codes for the amino acid alanine. The genetic code is always universal. It means that all known living organism systems use nucleic acids and the same 3 base codons, otherwise called the triplet codon, directs the synthesis of protein from amino acids. So the genetic code is universal. For example, AUG is the codon for methionine in mitochondria. The same codon AUG codes for isoleucine in cytoplasm. With some exceptions noted, the genetic code is universal. For example, the messenger RNA UUU codon codes for phenylalanine in all cells of all organisms. Some exceptions are reported in prokaryotic, mitochondrial and chloroplast genomes. However, similarities are more common than differences. Now, what is an overlapping codon? Means the same letter is not used for two different codons. For instance, the nucleotide sequence GUU, GUC represents only two codons. Here you can see an overlapping codon. The codon is AUU, GCU, CAG. Letters overlap. AUU, the same letters are used in the next codon, UUG, UGC. Now, in non-overlapping code, you can see, which is given below, AUU, GCU, CAG, where the alphabets are not repeated. It is a comma-less, which means that the message would be read directly from one end to the other. That is, no punctuation is needed between the two codes. Now, what is a degenerate code? It means that more than one triplet codon could code for a specific amino acid. For example, codons GUU, GUC, GUA and GUG. These codons code for valine. The genetic code is degenerate with many amino acids specified by more than one codon. The only amino acids like tryptophan and methionine are coded only by single codon. We must remember this always. Non-ambiguous code means that one codon will code for one amino acid. The code is always read in a fixed direction, that is from 5' to 3' direction. This is called polarity. Here you can see the messenger RNA codons. GCU codon 1, it codes for amino acid alanine. ACG codon 2, it codes for threonine. GAG codon 3, it codes for glutamate. CUU codon 4, it codes for leucine. CGG codon 5, it codes for arginine. AGC codon 6, it codes for serine. UAG codon 7 is a stop codon. What is the initiator codon? AUG has dual functions. It acts as initiator codon and also codes for the amino acid methionine. Here you can see the start codon AUG. In DNA, a start codon initiates the translation of the first amino acid in a polypeptide chain. Here you can see the messenger RNA with the multiple codons. AUG is the first codon that codes for methionine. GGC is the second codon, codes for glycine. UCC is a codon 3 with codes for serine. AUC is codon 4 that codes for isoleucine. GGC is a codon 5, codes for glycine. GCA codon 6 codes for alanine. UAA, which is the last codon, codon 7, is a stop codon. All these amino acids are linked together to form a polypeptide chain and linked by peptide bonds. And this is the primary structure of a protein. Now, what is the termination codon? UAA, UAG, UGA are codons. These are designated as termination codons, otherwise called stop codons. They are also called nonsense codons. Here you can see the start codon, AUG, and the stop codon, UAA, UAG and UGA. All these three are called stop codons. Whenever this codon is encountered, the translation stops. Here you can see the incoming transfer RNA, which is bounded to the amino acid. According to the anticodon, it matches with the codon on the messenger RNA, and the outgoing transfer RNA, which is empty, which does not contain the amino acid, and the amino acids together are linked by a peptide bond and they form a polypeptide chain. Now let us move on to mutation and the genetic code. Sudden change in a gene causes mutation. So comparative studies of mutations and corresponding alteration in amino acid sequence of certain proteins have confirmed the validity of the genetic code. What are the different types of mutations? Missense mutation. Mutation changes the type of protein made. Nonsense mutation. Mutation causes premature stop in translation. Silent mutation changes bases but has no effect on the protein made. Point mutation, change in one base along the DNA sequence. Substitution, replaces one base with a different one. Insertion, add one extra base into the sequence. Deletion, remove one base from the sequence. What is frame shift? Any insertion or deletion mutations shifts the bases. So this shifting of bases causes change in the amino acid sequence and the change in the formation of the proteins. Here you can see a normal DNA. with the codon TAT, which codes for tyrosine. CAT codes for histidine. CCT codes for proline, AAG codes for lysine, and GTA codes for valine. In substitution, that means the third codon, the C is substituted by G. Because of this change in the codon CGT, the arginine amino acid is linked in the protein instead of the proline. Now going to the insertion, where the G is inserted between C and C. Because of this, there is a shift in the coding towards the right side. And the third codon, which codes for proline, instead of that, arginine is linked. And the fourth codon is a stop codon. So that stops the synthesis of protein. And the next, it continues the synthesis where the GGT codes for glycine. In deletion, where the CAT, the A is deleted. Because of that, there is a shift in the frame towards the left side. So for example, the TAT codes for tyrosine, CTC codes for leucine, CTA codes for leucine, and AGG codes for arginine. The relationship between genes and the DNA are best understood by mutation studies. The simplest type of mutation at the molecular level is a change in the nucleotide that substitutes one base for another. Such changes are known as base substitutions, which may occur spontaneously or due to the action of the mutagens. A well-studied example is sickle cell anemia in humans, which results from a point mutation of an allele of beta hemoglobin gene, beta HB. A hemoglobin molecule normally consists of 4 polypeptide chains of 2 types, 2 alpha chains and 2 beta chains. Each chain has a heme group on its surface. The heme groups are involved in the binding of oxygen. The human blood disease sickle cell anemia is due to the abnormal hemoglobin. This abnormality in hemoglobin is due to a single base substitution of the 6th codon of the beta globin gene from GAG to GTG in the beta chain of hemoglobin. Here you can see the point mutation sickle cell anemia, where the RNA normally should form GAG in the codon, whereas instead of GAG, GUG is formed as the codon. Because of that, the normally GAG will code for glutamine and a normal protein is formed. Whereas in sickle cell disease, the A is replaced by U and this codon GUG codes for valine, and this forms a mutant protein. It results in a change of amino acid, glutamic acid to valine at the 6th position of the beta chain. This is the classical example of point mutation that results in the change of amino acid residue glutamic acid to valine. The mutant hemoglobin undergoes polymerization under oxygen tension, causing the change in the shape of the RBCs from biconcave to a sickle cell shape. Here you can see the messenger RNA that forms from the DNA results in the translation of glutamic acid normally, whereas in sickle cell disease, the translation of valine occurs in the protein. This results in the sickling of the red blood cells. Now, to understand the point mutation, the effect of point mutation can be understood by the following. You take the codons A, B, C, first codon, D, E, F, second codon, G, H, I, the third codon, and J, K, L as a fourth codon. If you insert a letter O between D, E, F and G, H, I, the arrangement would be shifted. So, it will form codon A, B, C, D, E, F, O, G, H, I, J, K and L is separate. If we insert O, Q at the same place, the arrangement would be A, B, C, D, E, F, O, Q, G, H, I, J and K, L is separated. Now, what is frame shift mutations? The above information shows that insertion or deletion of 1 or 2 bases changes the reading frame from the point of insertion or deletion. Such mutations are referred to as frame shift insertion or deletion mutations. This forms the genetic basis of the proof that codon is a triplet and is read in a continuous manner. So the examples of frame shift mutation, here you can see the normal sequence. This will change the entire DNA sequence, thus changing the entire amino acid sequence. This can be lethal, whether insertion or deletion of the nitrogenous base in the middle. For example, if you take the normal sequence ATC, GAT, TTA, AAG is the normal sequence. So the A is deleted and the rest of the bases will shift towards the left side. The mutated sequence will be ATC, GTT, TAA and AG. This will result in formation of mutant protein. Here you can see the addition mutation, where there will be addition of C between T and G. So TAC, TCG, TCT, AGT and ACA. Instead of the above amino acids that are formed, there will be altered amino acids formed. For example, instead of tyrosine, tryptophan, leucine, valine and histidine, the polypeptide will contain tyrosine, serine, alanine, serine and threonine. Now, what is Wobbles hypothesis? It is a hypothesis proposed by Crick in 1966. It states that tRNA anticodon has the ability to wobble at its 5' end by pairing with even non-complementary base of messenger RNA codon. In 1996, Crick proposed the Wobble hypothesis to explain the degeneracy of the genetic code. Except for tryptophan and methionine, more than one codon direct the synthesis of one amino acid. There are 61 codons that synthesize amino acids. Therefore, there must be 61 transfer RNAs, each having different anticodons. The total number of transfer RNAs is less than 61. According to this hypothesis, in codon-anticodon pairing, the third base may not be complementary. The third base of the codon is called wobble base, and this position is called wobble position. Here you can see the transfer RNA, the third base in the anticodon, which is called the wobble position. The actual base pairing occurs at first two positions only. The importance of wobbling hypothesis is that it reduces the number of transfer RNAs required for polypeptide synthesis, and it overcomes the effect of code degeneracy. Here you can see the normal pairing where GAG pairs with CUC, and this codes for amino acid leucine. In wobble pairing, there is CUU, which is a codon which codes for anticodon GAG, and the same brings amino acid leucine. In the above example, we have seen though the codon and the anticodon do not match perfectly, yet the required amino acid is brought perfectly. This enables the economy of the transfer RNAs. For example, GUU, GUC, GUA and GUG code for the amino acid valine. So today in molecular genetics part 6, we discussed about genetic code, mutation and the genetic code, and Wobbles hypothesis. Thank you. Kindly subscribe, like, share and comment the channel ReadMed Prep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is ReadMed Prep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.